Welcome to this model. I'll be talking about retrieving biological data or something like how to search in the nucleotide sequence database and aligning sequences and comparing sequences. And to start with, the biological databases were originally meant for storage of large sequence data and the genome data. But as we move on to the technology age, new algorithms got developed for sequence analysis and sequence comparison. So these algorithms built on top of this database to analyze a lot of sequence features. So for example, NCBI, EMBL, DDBJ provide all these kinds of features to you. Apart from that, there are some genome-centered databases, Ensemble browser, UCSC also provide these kinds of analyzing tools. And regarding protein databases, you have like protein sequence database like UniProt, then 3D structured database as like PDB, and the EBI also provides lots of sequence information. So this is the resource where what the European Bioinformatics Institute provide. They provide information of the DNA, RNA, gene expression data, and also the protein sequence data like families, motifs, and domains. Apart from that, it also provides the systems of the biological systems data like the reactions and the interactions and pathways which is going on, and the metabolomics data, and also something ontologies and literature, and also there are lots of other tools for doing all the bioinformatics and cheminformatics work. You can also access the data using APIs, and you can go to ebi.ac.uk/services where you can programmatically access the EBI resources. So Ontrans, it's from the US side. They also provide the same kind of thing like literature searching, doing sequence comparison, and doing searching sequences in large genomes, and searching proteins, and find curated data. It's a service from the NCBI and contains like 40 core search pages interconnecting different kinds of biological chemical information. So more resources like the GenBank is a sequence database that provides annotated collection of all genomic and the cDNA sequences and mRNA and RNA, all kinds of sequences in public domain. You also heard of the PubMed, which is a literature search engine, provides access to all the published articles on the journals of scientific interest. You can go to the YouTube videos for NCBI and LNM. They also provide the Facebook page where you can subscribe and get a lot of information about the tools what they provide, and for efficient searching, and for um, uh, development of other tools of NCBI. Another database is Ensemble database. It's a joint product from the EMBO and the Sanger Institute at Cambridge, it's, and it contains information about the phenotypic data, the gene expression data, and also it contains the latest ENCODE data. Apart from that, there has like highly carefully annotated manual curation of the human, mouse, and the zebrafish, pig, and dog databases. You can download most of the data from the website, and also they provide YouTube videos. You can go to the links which I provided here to see how the ensemble database works, and how does it look like. So the nice feature is kind of custom will keep tracks of this. If you do a search, so for example some gene, and you can, if you access it from other computer, so it keeps the history of the searches what you did, and it will help you to track down what searches you made in the past, and help you to identify uh, and help you to identify those searches. So basically it keeps the track of the searches that you made with other computer. And also, like you can see here, we were provided a picture of the chromosome, shows the chromosome look when you click this chromosome, it shows you a lot of like nice figures of the sequence contigs and the exons and the introns and the locations, and it's pretty informative site, and a lot of people use this site for doing high-end bioinformatics. Another site is the UCSC Genome Bioinformatics site. It's allowing for visualization of many genome features like mapping and sequencing, phenotype and disease associations, gene predictions, gene expression data, and also helps do some comparative genomics. So a tutorial is also provided at the website below if you want to go into deep of the UCSC Genome Bioinformatics. So you can just how the browser works. You can go there and see the tutorial. So the important facts about working with a sequence database is that, for example, in CPI, you can in the lab you can just generate sequence information and submit it to NCBI. But whether the submitted sequence is accurate or not, NCBI doesn't check any information about that. So the good thing is the ensemble checks these annotations and keeps track of what's good and what's bad. So NCBI doesn't provide all these things to you like the annotation. So anyone can submit sequences to NCBI and so that's why the development of Ensemble and other high quality databases came up so that it provides much uh, better information and it is also like curated. So annotation of the most of the genomes are done by researchers except for like specific reference sequence ref6. So these are like quite not curated by the genbugs in experts out there. So it means like if my lab generates a sequence information of one gene and other lab is generating the sequence information of the same gene, and we might both have different methods of generating the sequence. So it means that that information and my information is not the same. So in that case, it's kind of difficult to identify which sequence to take and which sequence to take for further analysis. And also, like all genomes are full of SNPs and other polymorphisms. So now gene finding algorithms are not perfect, especially when it comes to predicting the splice sites of the genes. And also, like alternative splicing can lead to different transcripts of RNA of the same gene. So there is no single correct sequence for a given gene. So these are the major drawbacks of this NCBI, and if you are not doing any annotation in the databases. So now I'll tell you if you want to extract some sequence information from the GenBank, how can you do that? So you can go to the GenBank at ncbi.nlm.nih.gov if you want to find some sequence regarding the E. coli TRPA gene, which is tryptophan A gene. You can just go there and just select the nucleotide down here and type the name, the Escherichia coli, and it will give you a lot of list of the standard GenBank entry, the genome databases, and you can also when you click one of these, you can just go to one of these pages and then just command F or Control F. You can just type like gene equals codes TRPA. It will just search in the whole page and give you the right positions, and then you just click the CDS. You'll get the gene. So this is how you can get the gene information from the whole genome of Escherichia coli. So here I provide some parts, some screenshots of the clicks. So here is this the TTTA is the gene information. It's the nucleotide sequence, and also at the top you can see it also shows the amino acid sequence. So the FASTA is the standard format for doing bioinformatics analysis, and you can just click the FASTA, and then it will convert you into the FASTA formatted file. And using that, and most of the softwares accept the FASTA formatted file. So FASTA formatted file starts with the GenBank accession number, then the chromosome number, then the name of the species, and what kind of sequence is that. It's a complete genome taken from the complete genome sequence, or partial genome sequence, something like that. Now coming to the homologous sequences, there are like we know that if sequences of two proteins are similar, they are most likely are derived from the same ancestor. and therefore they have similar structures and similar biological functions. So this is the main concept of the homology. So it means that if something is true for one sequence, DNA or protein, it's probably true for the other one also. Studying a protein, so studying a protein or a gene takes a years lot of time. So that's what the databases were developed. So if you find a sequence, you search, give a search to the database, and it will find you a lot of relevant sequences based on your query sequence. And also, like, if the sequences which come up in your list, so it means that proteins that have same ancestor might have, like, same ancestor or might be of from different species, so we can relate it, this protein is from this ancestor, and they might be homologous. So homologous have similar sequences and structures and functions. So they might have similar domains, similar motifs. So as a general rule, like, two proteins are likely to be homologous if their sequences are more than like 25% identical. So I mean identical means they have similar like A adenine adenine guanine guanine. So they should be identical. DNA sequences are homologous if they are at least 70% identical. So the protein sequence need to be like 25 and DNA like 70%. So don't confuse homology with similarity. Homology is a binary, like, is it homologous or non-homologous. So it reflects homology reflects the evolutionary relationship between the sequences. Similarity is a quantitative measure. Two sequences can be more similar or less similar. So similarity between two sequences doesn't reflect that whether they are evolutionary related. Okay. So then the sequence similarity comes like a pairwise comparison of sequences is very common task which is done in bioinformatics and like in genomics, comparative genomics, analyzing genomes of two different species. That while doing this sequence similarity, we align the two sequences and test the hypothesis that the sequences are homologous or not, like meaning that each pair aligned residues are descendant from a common ancestral point or root or not. If two sequences have some similarity simply by chance, this does not have any biological significance. But if two sequences are homologous, by comparing them, we can gain information about the structure, function, and evolution. So for example, computation alignment is done. I have an example on the next slide. So for example, you are aligning two sequences A, these two sequences I've mentioned. So for a match, I give a score of 3. For a mismatch, I give a score of -1. And if I introduce a gap, I'll give a score of -5. So we'll be doing some sequence alignment coding in the following lectures, where I will show you how to code this algorithm and how to find the score of two aligned sequences. So here you can see, like, there are 1, 2, 3, 4, 5. In the first example, it shows there are like 5 matches. So, and also there are like 3 mismatches. So the course optimal score comes around into 12. So now in the next sequence, you see when I put gaps, it comes around +7, and then more gaps can be introduced, and comes the -10. So the choice of parameters depends on the biological model, the choice, and you don't. And the good thing, there should be an optimal alignment between the two sequences. So to do this, you need to do the dynamical programming, which will help you to optimally align the two sequences with the score. Mm, So basic alignment search tool is a very popular tool to do sequence comparison and search. Another is the FASTA. So they are the right now they are the uh, tools to doing sequence comparison. You can go to YouTube to see what are the BAST and the FASTA programs, and there's also a tutorial of BLAST and the PS position specific iterated BLAST, which actually iterates every time the sequences get searched in the databases. You can get the tutorial down on the YouTube. So BLAST outputs some bit score value, and it indicates how good your match is. So your score is like below 50, just you need to ignore that. Also, it comes with the e-value, which shows the significance of the match to the hit. So once you give a query, it will search the whole databases of sequences and align them and gives you a score along with the e-value. And the e-value indicates statistical significance, so which indicates how much likely this match would be found if there is no real match in the databases. So e-value like below 0.001 is worth exploring, and actually up when the e-value is like 0, it indicates a very perfect match between your query and the database sequences. So there's some. I have also provided the PPT as in SlideShare. Apart from that, you need to go to this the UniProt. How to search protein sequences in the UniProt. You should go through this tutorial, and there's also the Reactome database, which is the pathway database, and how the pathways are like this information is stored, how you can get information is all these things is provided. And also this 3D PDB 3D structure database, the PDB is also there, where you can get the 3D structure of the proteins, and from different species, and also get the ligands which attach to these proteins. It has this uh, the 3D structure gets information from the X-ray crystallography and the NMR. I'll also give a link to the Farm GKB, which shows you the SNPs, the ethnicity, and the human genes, and the information of the drugs. So it shows the effects of the drugs of different ethnicity, and dose information and all these things. So explore this page, and thank you.